Good. And then everyone can see my screens, correct?
Yep.
One screen, one slide, we're good.
Yep.
Great. Thanks, everyone. So thank you for the opportunity to come today and to talk to you a little bit about something that I really love and that I've been working on and still in this field as of today is my career. And hopefully we can discuss just how to jog your memories on or jog your just interest in molecular biology and get a project or something that you might be interested in to work for the MCEF Fair. So as Becca has already introduced me a little bit, but I'll introduce myself, and I even put a little picture on there just in case, you wanted to see another form of picture of me. My name is Dr. Nicole Barkley. I have a PhD in biology, and I focused on cancer biology, and I currently work at a global pharmaceutical company on cancer clinical trials. And as mentioned, I'm a previous MCEF mentor. So I had a mentee that did a project with MCEF, and we'll talk a little bit about what she did and how she worked through that and used molecular biology techniques. And then I'm also a current board member. And I did want to show a little bit of what I used to do in grad school and why I love cell biology and molecular biology is because my focus was on prostate cancer development. And that involves a lot of molecular biology techniques. And one of them that I have in these pictures shown here is called immunofluorescence, which is basically lighting up proteins on a cell. And one of the pictures here on your left-hand side shows what the protein looks like, which is in red, when everything is regulated correctly in a prostate cancer cell. The protein is located in the location that it's supposed to be. It's outside in the cytoplasm, and then also within the nucleus, which is this blue colored circle thing right here. But when we unregulate the protein in some way, shape, or form, I mutated the protein so that it didn't go into the appropriate location. It all localized within the nucleus and it didn't come out where it wanted to be in the cytoplasm. And so this is just one of the reasons and things that we can talk about and, kind of techniques that I use, but you could definitely use something more simpler than this, but something definitely cool to look at and why I'm so interested in molecular biology and cell biology. So what today we'll talk a little bit about what molecular biology is, and then two topics within molecular biology, which is the central dogma and cell cycle. Then we'll talk a little bit high level on the cell scientific method, and then I'll walk through those individual steps in the scientific method and how you can start to probe around what kind of molecular biology project you might want to do and how to apply different techniques into that project, and then give that example of an MCEF project that was done. And there was two parts to that project. But I'll go through just part one. And then I'll also at the end kind of like high level some topics that I find interesting that if I was in high school or in middle school, I would want to do. And maybe that will give you some ideas of things to ask or think about after today's talk. And by the way, just stop me if you want to at any time. I don't know if that's okay, Becca, if we can have questions within, but let me know if there's ever a question on anything that's discussed.
Yep. I'll keep an eye on any hands up and I'll keep an eye on the chat. So, but folks.
Great. So what is molecular biology? It's a branch of biology where we're focusing on understanding structure, function, and interactions of biological molecules. Very high level, really broad definition, but we're going to drill that into what kind of biological molecules are we looking at and focusing in molecular biology. And most of you probably have heard of DNA, right? That is a biomolecule. So deoxynucleic acid is a biomolecule. But there's also another one called RNA, which is ribose nucleic acid. And then there's the functional aspect of those biomolecules, which is the proteins. And this is a different type of protein than chicken and steak and maybe the fish you probably eat. But these are the functional proteins that help move things in your body and make sure that you're alive or that you can breathe or that you can move your arm when you want to or that you want to run, you can. And so there's all types of pieces of these biomolecules in your system currently, and they're working to continue different life processes that are going on inside. So what is one of the things about molecular biology is actually understanding how these biological processes and these actual biological molecules function to allow for life processes to continue to go. So some of the things we'll talk about is a little bit about how we inherit our genes, right? Inheritance, how genetic information is stored and passed between generations. Or as I said before, protein synthesis, like how your functional parts of these biomolecules actually become a thing and then support your cells as they do different types of processes within your body. And some of the things that these biomolecules can interact with is called the cell cycle, and we'll talk a little bit about that in a minute. But they also can affect how diseases progress. So that's the other thing, right? So even though there's a natural and healthy and normal way of how these biomolecules work, there's also a way where they're deregulated or unregulated and they cause diseases. And this is something else that is studied a lot in molecular biology. So when I said earlier that I would talk a little bit about central dogma, I wanted to talk about how this plays a role in molecular biology, and then we'll talk a little bit about cell cycle. So the central dogma, it says is a theory. They keep saying it's a theory, but technically it's almost a proven theory at this moment. But we consider this a theory that states genetic information flows in one direction, from DNA to RNA to protein. And we talked about the fact that those are all the biomolecules that are involved in molecular biology. And to break it down even further, DNA, as we all know, is the molecular or biomolecule that stores all of our genetic information. This is how we get instructions on making those functional biomolecules, which are those proteins. So here in this image here on the right-hand side, you'll see that you have this DNA molecules and a double-stranded DNA molecule, or as we all probably know as a helix. And the next part of this is a process called transcription, or how we make more of that, not make more, but sorry, how we read the DNA into RNA. And so RNA is basically the messenger. It takes that information that's held in the DNA and now it makes it into a message that can be translated. And that's another process. So how you translate from a language to a different language, very similar. The RNA is that intermediate step between DNA and that functional biomolecule that makes that protein. So RNA will be the character that reads the DNA, gives the information, and to ribosomes, which are part of this situation, which is not in this image, but there's a translator called the ribosome that translate that information from RNA into proteins. And those proteins are that functional molecule that I keep mentioning. And they perform a lot of the tasks that are involved in your cells today. And they basically take that information that was used from the DNA, read by the RNA, to make those functions go. Any questions on the central dogma so far? So how does this play a role in cell cycle? And let me just first tell you that the cell cycle is my favorite thing in the whole wide world. I'm just going to let you know up front, because everything revolves around how your cells are dividing and growing and dividing and growing. And it just basically gives us the framework of how we get into the cell cycle and how we have multiple cells start to go and divide. So DNA, RNA, and proteins are essential molecules for the cellular functions in the cell cycle because it includes helping the cell to grow in order for it to get ready for division. It allows for the DNA replication because each of your cells all have the same DNA instructions within the cells for certain cells. So yes, you need to have DNA replicate in order to do your cell division. And then you have the actual splitting of the cells, and so that you can get more than one of those cells. It's such a tightly controlled process that it even has checkpoints, and those checkpoints are regulated by functional proteins. So there's the G1, G2, and a metaphase checkpoint, and it wants to make sure that at each of those checkpoints, the process that are happening in those different phases finished and was accurate. And those are those checkpoints. They make sure everything is going correctly. So even though there's like here, you see in this image on the right-hand side that it's about 4 processes within, there's this initiation process called the GO process, where it starts off like, we know we need more of this cell. So now we go into, we need to make this cell bigger. That's that cell growth phase where it sees G1. Then it goes into the S phase, which is the DNA replication, because you want to make sure you have all of the DNA that you need in the next cell as well when you do your division. You do a preparation for that cell division, and then you undergo the M phase, which is that cell division phase. But these checkpoints are super critical and ensures that the cell maintains healthiness, that the DNA is correctly replicated, and everything is aligned as far as your chromosomes before the cell divides. And this makes it ensure that everything is healthy in the cell cycle. So again, making sure that that DNA, RNA, part of those molecules is also part of this cell cycling piece as well. So if you don't realize how important cell cycle is, it's super important because even though it's tightly regulated, what happens is that when it becomes unregulated or dysregulated, this is where some of our diseases can occur in that cell cycle, which is why it's super important to understand all of those mechanisms and those biomolecules that function in the cell cycle. So we all hear about cancer a lot, right? And one of the things I work on a lot is in cancer. And one of the leading causes of cancer right now is genetic mutations, right? Genetic mutations that deregulate that cell cycle process. And sometimes what it deregulates is that growth phase, that just uncontrollable growth and uncontrollable division. So it doesn't check to make sure that the DNA replicates correctly. It just keeps growing regardless. It could have been errors within that DNA replication, but there was no checkpoint because of the unregulated cause or some mutation that caused the cell cycle checkpoint not to regulate that. And so instead of it stopping the cell in its tracks, it kept going. And this is how usually most tumors form. Other diseases like autoimmune disease can also have a defective cell cycle where you either have excessive division without stopping, or you actually even experience some cell loss where you're not getting enough cell division. So those can be another way that cell cycle deregulation affects diseases. And then even cardiovascular diseases. Some research looks at just overall heart attacks being caused by the loss of cells and not having the appropriate cell division occur. So again, lots of diseases is a tightly regulated system. And when it works well, everything goes well. When it's deregulated, then you have all types of diseases that can form from those deregulations. So molecular biology has a lot of different things that you can start off with, but understanding those two major two major topics like the cell cycle and the central dogma is crucial and important in starting and understanding how to use molecular cell biology. And I'll pause there before we go to the next topic because we're going to go into the scientific method. Is there any questions so far? Are we all nervous to ask a question? No, we're good? Good. So I'm going to walk a little bit through how I look at the scientific method. You probably heard this several times over again, and I'm sure you've heard it in school, but I have four main steps that I like to look at in the scientific method, and I'm going to outline them in each step within the PowerPoint presentation, as well as how you can think about within those steps how to develop a molecular biology project. So the four steps I like to look at with the scientific method is one, observation/research of a topic. The second step being form a hypothesis or a question. 3, overall conducting the experiment, and we'll walk a little bit through those details of how to conduct the experiment. And then step 4 is your analysis of the results, and reporting out those results, which would be, in our case, we would focus on going to MCEP and reporting out your results, or even doing a presentation in school and doing your results, or even going to a smaller conference within your high school or your state and actually reporting out your results. So you want to always, as a scientist, one of the biggest things to do is to report out what you've done and look at your research and be able to explain to others how you did it. So if we go through step 1, and here's some of just high-level pro questions that I wanted to walk through with everyone is your observation, your question. So what I like to look at when I'm looking into a project or even thinking about a project is what interests me about that particular topic. So question you could ask yourself is what interests you about molecular biology? And then clearly state what you want to investigate when it comes to molecular biology. So maybe you're interested in, now we talked about cell cycle, how genetic information is passed down between generations, right? It could be something as simple as how certain types of traits get passed down from generation to generation. And what does that look like? And how would you test something like that? Or how do cells interact? So we talked a little bit about the cell cycle, right? How do they interact in an organism? Do they all interact the same way? Does your skin cells act the same ways? Maybe your cells in your liver, maybe not. I can guarantee you they don't. But they still have their own function and they have their own biomolecules. And there's an important process called cell signaling that may be of interest to you, and maybe something that you can use a model organism to assess and to look at a little bit more to understand. Or how does DNA play a role in diseases such as sickle cell anemia? And this is where you start to think about those different types of questions, those different things you're interested in. And then you go in and you start looking at publications, or you start talking about topics that you thought about through in class, or even talk to a mentor or a teacher. And then, or you could even look at topics that affect the world right now. I know a lot of things are going around about vaccines, something that looks in interest to you. How do you start to probe a little bit more into those different topics? And then start doing some of that research to understand what are the holes or the gaps or things or questions that you wish you knew or understood that you can't seem to find in those research topics. And that's where you start to probe a little bit into what your hypothesis could be. So once you identify whatever those interests are, then you want to start looking into what your potential question is and how you want to support your investigation. So this is where you start looking into your hypothesis and your question. But one of the major things about a good hypothesis, it needs to be testable, right? So whatever you come up with your hypothesis, it must be possible to design an experiment and observe that result, right? And one thing I wanted to note, and one of the best things about science, and everybody doesn't talk about that, positive results are always great, but negative results also are great too, because it gives you some information about maybe this was a good idea, but now I have to think about this in a different way because I didn't get the result that I really intended. And what does this result tell me? So not only proving your hypothesis is good, but also maybe disproving your hypothesis is also a good thing to do, and also worthy of being reported out. You want to do a lot of your, again, you want to make sure your background information and your research is good. You want to make sure you have, I call it an educated guess, not a random guess, because again, you're wanting to test your theory here. And so it might be a guess, but you want to make sure it's based off of, information that's already out there and data that you can point to. You want to make sure it's specific and clear, super important. And you want to make sure it very, very clearly states your relationship between your independent variable and your dependent variables in your study. So I put some examples of what a hypothesis could be. Here I say, if we activate the gene, and I'm throwing in the cell cycle since we talked about the cell cycle a little bit earlier, known to be involved in the cell cycle regulation, then we will observe a significant increase in the rate of cell division in a laboratory setting. So that's my hypothesis. Let's say that is like I increase some type gene in the cell cycle, and I want to know if that increases the rate of cell division. Then the rationale behind this is the hypothesis based on background information or knowledge that certain genes directly control cell growth and division. So this could be me saying, hey, I know that there's this checkpoint genes out there. Maybe if I change this, this will increase cell growth. And then you could also parlay that and say, well, that's interesting because I know cancer is uncontrolled cell growth. So how does this relate to diseases that I can actually point my research to and make it sound really, really super interesting when you start thinking about how you can relate to current status or current topics of the world today. So just an example, something to think through about how to form a hypothesis. Did I go backwards? I did, sorry. So step 3, which is a longer step, and I like to go through this a little bit slower because you want to make sure when you're conducting your experiment, you're planning out the details, especially with molecular biology. It's can be super complex sometimes, depending on what you're going to do. You don't want to miss a step, and you don't want to forget to do a measurement or forget what you wanted to do afterwards. You want to make sure you have a good plan outlined and written and ready to go. So make sure you're outlining all of your steps. You know, maybe at the beginning, you don't know everything you need to do, but you want to know a high level of what you want to do. So you outline your steps, you include the measurements you want to think about taking. You want to look at your variables you want to measure, and you want to also identify any potential methods that you're interested in. And make sure you do any type of research to understand how to do those methods. Once you select your methods, whatever they may be, depending on your topic, choose the appropriate way to do it, right? So make sure you have the equipment or the resources to do it before you finalize your protocol. And then you also want to make sure you have your appropriate controls. So you want to have a negative and positive control for sure, because you want to make sure that when you assess and analyze your data, you have help in making sure that it wasn't because of this thing, but you had that positive control that said, yes, this worked well. Negative control also worked well. So you can really know that your changing of your variable is what caused your result. If you need replicates, and I always say replicates are also a very optional thing depending on your program, depending on your project. But one of the things that allow for replicates is to give you more statistical power in your analysis as well. So you want to have more than just one replicate, right? You want to have at least 2 or 3 replicates so that you can say you have a powered enough analysis to say that multiple times you saw the same thing occur. Because always when I say research, it means repeat, right? How many times you repeated it and were able to confirm your results. And then one thing I like to do, and I still do to this day, is write out a detailed protocol. Make sure you have it listed out. So now you have your outline, you know what you're gonna do. Write this very, very detailed protocol. Step 1, this is what we're going to do. Step 2, this is what we're going to do. And you make sure you know what you're gonna measure to each of those steps and keep it controlled so that you're not missing anything. And actually get somebody to review it and make sure that you're not missing anything when you're thinking through it. That's also very helpful, and make sure that you don't forget anything while you're conducting your experiment. Once you get all that done, you prepare your materials, make sure you have all the necessary requirements that you need to do to conduct your experiments. And also then you follow your procedures, your detailed protocol, and you make sure you write out everything you observe. Even though you don't think it's really relevant, it may be relevant at the end of the day when you want to do your assessment of the data. Any questions on that? I know that was a lot, but I liked, you know, doing experiments, you want to make sure that you keep it very controlled and very organized so that you don't miss anything.
I think that's great, Nicole. I think also the emphasis on making sure everything's really well planned out is also essential when you're submitting paperwork to be approved. So
That's also very right, because that's right. You
right.
do have to do a lot of the improvement of the paperwork. So it's kind of killing 2 birds with 1 stone here, right? Making sure it's already organized and you still have to submit it anyway. Yep.
Yep.
And then you analyze and report out your results. So here it depends on your program of approach. I keep saying program, apologies. Look at me listening to my work. But depending on your project, you can use graphs, you can use tables, you can use very high level basic statistical analysis to analyze your data depending on what you're going to do. And then you want to look at your data and interpret your results, right? So you want to draw some conclusions, you know, determining whether or not you proved your hypothesis correct, or even if you disproved your hypothesis and why. And you want to think through how you want to conclude your data. And then of course you want to present that study. So choose the best way to illustrate. You know, I know for MCEF it's a lot of posters. It's some of the students even brought like models to show us how they did their studies. So the best way to show your results and present it for any type of research discussion will be solely up to you, but you want to choose the best way to illustrate what you did. So those are the 4 main steps with that. And before I go into just the example, is there any other questions that you had on just the scientific method and maybe how to jog your memory for the molecular biology piece? If not, I'll go into my mentee's example and I'll thank my mentee, Oge. I think she actually was in part of a panel this last weekend, which was awesome.
Yep.
She didn't even tell me that. I called her. She never even said she was
I'll
doing this.
send you a picture. Yeah.
I know. Thank you. Really cool student that I met when I was a MCEF mentor, and she came to me with a very cool project, which, blew my mind, but she did two parts to this project. I'm going to walk through part one, where her interest lied from, and how she decided to use some molecular biology techniques to get this project going. So um, Oge was interested in epigenetics, which in high school I thought was really crazy, but she was she was very interested in epigenetics. Um, and I told her, I was like, "Do you know what the epigenome is?" And she was like, "Not really." And so she went and did some research, and she realized it was, epigenome regulates the expression of the genome, right? That DNA. And it does this by altering its availability to transcription factors. So basically it makes it harder for either it to be transcripted or replicated or anything. But those are things that happen just within your body. So it's a cool kind of other system in your body that I don't even talk about, but it's also molecular biology related. And so she did a lot of research, and what interested her about the epigenome was this paper she read about the Holocaust survivors. So she read a paper that looked at a study where Holocaust survivors revealed that parental PTSD from the Holocaust influenced the epigenome of their offspring, and that altered the offspring's psychology, therefore increasing the likelihood of developing mental disorders. And so that's what she started from, that paper. And so now she wanted to understand, how does parental PTSD changes through generations, and determine if epigenetic changes can be reversed? So that was her question, basically high level, her hypothesis. Um, what she did was, we talked a lot. I will say this took a long time to figure out how we were going to do this. Um, but ultimately, she wanted to use a model organism. And what I told her was like, you want something that's quick, cheap, and cost effective for you, right? It's something that you don't have to manage too much while you're in school. So we picked Drosophila. And Drosophila are a very versatile animal. They're used in behavioral studies and genetic studies all over. There's tons of papers using Drosophila. But one of the things that make them ideal for testing something like this is that they have a short life cycle. And so it's easy to study and easy to get information and look at different generations really quickly without waiting for, a mouse to mate and then have babies and then them grow up. By that time, MSF would have been over. So we needed something really quick in order to look at multiple different generations really fast and get some data to generate. So this is what we decided, and we did some research on how to use this particular Drosophila hydae in the study. And then,
I will say, for those that don't know, that Drosophila is also known as fruit flies.
sorry, that's a great thank you for adding that.
Yeah.
Yes, they're fruit flies, and they can be annoying. So before you start that, make sure you get permission from your parents, because they could fly around your house and you don't want that too much. So make sure, but they can be annoying. And Oge also did some research on methods to use. And so she found two different types of methods that could use to assess stress and anxiety. One was called a wall following behavior assay, and then another one was called a larval locomotion assay. And with each of these assays, the goal was to see if she stressed out the parental generation and they ended up mating, did their children, their F1, which is the children generation, have issues with movement and following the wall, which was actually part of what these Drosophila, this specific brand of Drosophila, was known for. So this was really interesting. She found out these methods and how they would work. And then she did her data analysis. So she used several different graphs to show like changes over time with parents that were stressed versus non-stressed parents. She stressed them using different types of holding out food from them or exposing them to heat for a certain period of time. So it was a very different types of stress. And then she, of course, she had her controls where she didn't stress those parents, and then saw what the F1 generation or the kids ended up being like. And she did look at some statistical methods that she got help from her teacher at school, but she was able to do a lot of analysis. And what she drew from her part one of her conclusions, that what she saw, what she wanted to see and hoped to get to, was that individuals would understand how wars influence their children and the traumatic events can affect overall generations. And the goal was that she saw that in her initial studies, that she did see that the effect of parents that were stressed did end up affecting how the kids saw stress and were affected also with the flies. And so she presented that and actually won. So it was a cool project. And she wanted to understand more about epigenetics. And one of the things she said in her future directions would be to know a little bit more about the DNA and how the epigenome played a role in that. And so that would be a future topic for her. But it all stemmed from that one Holocaust paper that she read. And then I wanted to add just before we end it, because I know I talked a lot, and I wanted to say some other topics that I would found interesting. I said genes to genetic diseases, what mutations matter. There's ways to look at this using plants even, because plants have a very similar cell division cycle and cell cycle, similar to mammals, but not, it has a little bit differences, but you could look at that and look at mutations. Can plants be resistant to diseases? Another cool thing that you could look at for molecular biology topics, and how you can try to mimic other techniques to see how plants can be resistant, and then even think through how you could use that for crops going down the future, right? So I know a lot of things about genetically modified crops are out there, but how do you make them resistant to disease? Um, you can look at pedigree analyses. We talked a little bit about inheritance. That's a really cool thing to look at, and you can use fruit flies to do some of that analysis if you wanted to. Or why do people respond to drugs differently? No, we all use different types of medications nowadays, and some people react differently than others. Is there a genetic or a cell signaling pathway that is different between generations, or even different between people, or different between men and women, or different between different ethnic groups, and things that you could think about and even start asking questions on, and starting to probe how you would start looking into those different high level topics. But I just also wanted to throw in a couple of things that are, if I were you, I would be interested in. And unfortunately, I can't do this anymore, but you could if you wanted to do a different project. But that's just it for me. I had a last slide of any questions, and I'll stop there and pause here. I hope I gave everybody enough time for questions.
Oh, you definitely did. I'm going to stop recording because I just want that section to be what we share, and I want folks to feel like they can ask questions.